So, ladies and gentlemen, dear colleagues, I'm very happy to be here at that situation, a special moment that we can restart the MENDEL lectures after almost 1.5 years by a next lecture. And it's my great honor to introduce Professor Andrew deMello. He's a British chemist and currently Professor of Biochemical Engineering at ETH Zürich. Andrew obtained a first-class degree in chemistry and PhD in Molecular Photophysics from Imperial College London, and subsequently held a postdoctoral fellowship at the University of California, Berkeley. Prior to his arrival to Zurich, he was a professor of chemical nanosciences and head of the nanostructured materials and devices section at the chemistry department at Imperial College London. Science originating from deMello groups has been recognized through a large number of international awards, including a Clifford Patterson Medal from the Royal Society of Great Britain, the Cordell Morgan Medal from the Royal Society of Chemistry, the Pioneers of Miniaturization Award, and the Advances in Measurement Science lectureship from the American Chemical Society. His group is engaged in area of microfluidics and nanoscale science, including the development of microfluidic devices for high-throughput biological experimentation and point-of-care diagnostics. So now I'm giving the floor to Andrew. He will tell us a bit more about this fascinating field, growing field, which is becoming a future of life science and medical sciences. So please welcome with me Professor Andrew deMello.
Thank you very much, Zbigniew, for a very kind introduction. It's fantastic to be here today, especially because this is really the first opportunity I've had in a very long time to give a lecture in person. And also just to be able to come back to Brno, to see lots of friends and colleagues, and hopefully also have a chance to talk a little bit about some of the activities that we've done with our collaborators. So the first thing that I would like to do today is really raise a confession, and you're going to find out very, very quickly that I'm not a geneticist. And so I feel a little bit of a fraud speaking to you. You'll find out as well that I'm not a biologist, and very quickly I think you're going to find out that I'm a technologist. And so today what I'd like to do is tell you a little bit about technologies that we've been developing in my group, initially at Imperial College and much more recently at ETH in Zurich, that really we hope will help the chemist and particularly the biologist to do different experiments. So I'd like to give you a little bit of a story about how I entered the field and the kinds of things that we've been doing with a particular relevance to biological problems. But what I'd like to do is really start with a slide that I'm sure no other MENDEL lecture has ever used before as a first slide of the talk, and you'll see very quickly why, and it's absolutely nothing to do with biology. So this slide here really defines what I would call really one of the two great revolutions of the 21st century, or the 20th century, excuse me, which was the Semiconductor Revolution. And you can see here 4 different individuals, and they made 2 very, very important devices. So these 3 gentlemen here were John Bardeen, William Shockley, and Walter Brattain. And just before Christmas in 1947, what they did, or 1948, is they created this very ugly device here. But this was really one of the most important things that was made in the 20th century. I'm sure many of you know what this is. We've got a bunch of wires. We've got a little crystal here of germanium. This was the first transistor. And what the transistor did is it allowed you to amplify, or it allowed you to switch a voltage very, very easily. And actually, although it doesn't look like it here, in quite a miniaturized form. So this replaced things like vacuum tubes and relays, which people had used in switches before. And it started to make people think about making complex electrical circuits. Now this chap here, again a very famous guy called Jack Kilby, was working at Texas Instruments around 10 years later. And he realized that he could take a lot of these ideas here and start to create transistors or other electrical components on a monolithic piece of semiconductor. So you can see this is an equally ugly device, but probably, in my opinion, the most important device that we saw in the 20th century. This is the first integrated circuit. And so what he did was he said, we can start to make these components in a monolithic way on a very, very small substrate. We can start to solve a problem that had really plagued electrical engineering for many, many years, and that was the tyranny of numbers. Although electrical circuits were really easy to make, as soon as you tried to make anything complex, all the wires and the components that you can see here really got in the way, and it stopped you from doing powerful things. So what this did was it changed the game. It started to tell us that electronic circuits could be really powerful, and we most easily see this through here, just a graphical representation of something I'm sure you've heard about before, which is Moore's law. And what Moore's law tells us, it's really a prediction, is that maybe here, if we look at the number of transistors on a processor, Gordon Moore, the founder of Intel, expected that that number would double every year to 18 months. And if you follow the next 50 years here, you can see that prediction has really been borne out. This is a logarithmic scale here. So back in 1971, 72, Intel's first chip here, the 4004, had about 2,500 transistors. The AMD EPYC Rome processor here now has around 50 billion. And this very, very simple fact, by making things smaller, improves performance. And so now, in a standard smartphone, we would have the computational power here maybe 10,000 times in excess of maybe one of the world's first supercomputers, which was the Cray 1 back in the mid-70s. So the very simple idea is that miniaturization allows us to do things better in terms of electronics. So what I'd like to do today is to try and convince you that we can use very similar ideas to think about improving experimentation, and particularly biological experimentation. So the kind of technology I'll talk about today, we can generally call lab-on-a-chip technology. More specifically, I'm going to be talking about microfluidic technology, but you can see really a very beautiful example. This is quite an old device now, made by Stephen Quake's group, initially at Caltech and latterly at Stanford University. And this contains a whole bunch of components which allow you to do things, in this case to bacteria. It allows you to do a complex chemical process or biological process in a very, very efficient way. So you can see here that what we've got here is something which looks a little bit like an electronic circuit, but now the components are going to allow us to do something which we would more normally do inside a laboratory. So what kind of things are we going to need to do? Well, this is a very generic slide. It kind of tells you the things that you need to do when you do a biological experiment. And I'm not going to go through things in detail, but you can see that we need to sample, get our sample from somewhere, maybe the lab or the environment or the body. We may need to pre-treat it in some way, get it into a form that we can process. We might then need to do reactions on it to form particular products. Unless we're very, very good, we then need to do separation and detection here. So we need to work out what we've made, need to work out how much we've made. And then in modern biotechnology and pharmaceutical science, we often would like to isolate and collect things. So the challenge here is, how do we do all this in a rapid, efficient, and robust manner? And that's a big challenge, because if you think of it, many of these processes, if not all of them, involve different instruments. They might involve different researchers or experimentalists. They may be in different locations. And what that means is this process can be very time-consuming. It's going to be normally costly. And at each stage here, we're going to introduce error. And so really what I would call the field of microfluidics, or as we just called it, lab on a chip, really started with a view to integrating all those processes, maybe on a monolithic device, and seeing how that would improve all of those figures of merit: cost, speed, efficiency. And so this slide here just shows you the kind of technology that I'm going to talk about today. So I'm not going to really go into detail about how we make these devices. We use very simple microfabrication tools, but you can see here examples of devices made in glass here, in polymers. This is an elastomeric polymer called polydimethyl siloxane, and here a device made in silicon, and these do completely different things. This is a device that allows us to do radiochemistry. This is a device that we'll talk about a little bit later, which allows us to manipulate single cells, and this is a device that allows us to do small molecule chemistry. And there are lots of different advantages that you can think about when you start to think about using microfluidic technology. So I guess my job today is to kind of give you reasons to use microfluidic systems to address particular problems. Now, they're not always going to be the solution, but they can often be a huge advantage. So these bullet points at the top here really tell you some very obvious things about miniaturization. If we can use microfabrication techniques, we can start to make things very cheaply. We're going to clearly use very small amounts of sample and reagent. And again, to give you an idea, for the devices and for the systems that I'm going to talk about today, we typically use volumes which are going to be sub-microliter and probably go down to the hundreds of femtoliter scale. So we're talking about volumes which are millions of times smaller than a pinprick of blood here. So we're very good at using sample and power. And then clearly we can think about making things portable. If we can miniaturize a lab down to a small device, we can think about taking it to different environments. So there are lots of other reasons why microfluidics is perhaps an interesting way to go. And these points here that I'm hopefully going to pull out in the next few slides, I think we can distill down into one statement here at the bottom. And I think this is one of the very best reasons why you should think about using a microfluidic system. And that's that we can generate, today's context, biological information that's very, very high quality, and we can do that very, very quickly. So in all the examples I'm going to talk about today, hopefully you'll be able to see that what we're doing is effectively doing experiments very, very well, and also doing them very, very quickly. So I don't get to spend any time on telling you really why microfluidic systems are so different, and I'm not going to go through many equations today, but I thought it would be useful just to highlight a few features that we're going to see, because fluids behave completely differently when you put them into a small, confined environment. They behave in a way that's very different to what we're used to. And so the first thing that we need to remember is if we take any component, let's just imagine a little channel, and we shrink it down and we make it smaller and smaller, our surface area to volume ratio increases. And so surfaces play a huge role on the microscale, but most importantly for us, they're going to mean that we can move mass, so that's molecules, and we can move heat around very, very efficiently. So we're going to be able to control how we do that, and we're going to be able to do it much more quickly than we could do if we were working on a large scale. Now what happens is as we also decrease scale, what we find is that viscosity or viscous forces really start to dominate over what we're normally used to, which are inertial forces. And I just want to explain that by showing you again a nice example here. Again, a very old study here by George Whiteside's group at Harvard University. What you're looking at here is a simple continuous flow microfluidic system. We're flowing solutions from the top down these channels here into a single main channel which moves down into the left-hand side. Now, the important thing to note about this experiment that the Whiteside's group have done is the solvent in each case is identical. I forget what it is, it's probably water. They've simply changed the dye that they've put into each of these streams. And you can see immediately that this is very unlike the behavior that we're used to on the large scale. Our flow streams move together in a very smooth, predictable manner. If we're going to mix these fluids together, we're going to have to use things like diffusion, which occur orthogonal to the flow. So this really mimics how high viscosity fluids behave on a large scale. But what that means for us is that things happen in a little bit of a different way. And I could spend a lot of time using nice dimensionless numbers to explain the behavior, but hopefully you'll see through example today how these features tend to give us very large advantage when we're doing chemical and biological experiments. So I thought a nice place to start would be really where I began microfluidics, and I guess I'm lucky or unlucky enough to have been involved in the field really from very early days. And this probably is the first example of what I would call a proper lab on a chip to do a complex biological analysis. And if you look at the date here, this is back in 1996. So this was really in the midst of the Human Genome Project, where sequencing of DNA was based on classic techniques like PCR and capillary gel electrophoresis. And so what I'm showing you here is probably a device that's pretty ugly like the transistor and the integrated circuit that I showed you earlier on, but shows how you can use integration. So this is a device which contained two components. This is a silicon microreactor, which is going to allow us to amplify our DNA via PCR. And then we have this planar glass electrophoresis chip. So this is going to allow us to take the products from that PCR reaction and separate them based on their size. So two classic molecular biology techniques that would be used in the lab. Now, when you do this kind of analysis using conventional technology, the process, certainly back in the mid-1990s, would take many, many hours, 5 or 6 hours to do something that I'm going to show you now. So using our technology, where we created here a micromachine silica microreactor, this was actually developed by Alan Northrup's group at Lawrence Livermore Labs, and integrating it to this electrophoresis device, the first thing that we could do was we could move heat incredibly efficiently. So you can see here just a profile of time or temperature here as a function of time. And as you all know, to affect a PCR reaction, we simply move between our denaturation temperature and a lower temperature to anneal our primers, and then a mid-temperature to extend that. And we repeat that process again and again. And what we can then do is simply use an electric field to pull some of that sample into our electrophoresis chip here, and then by applying an electric field, we can then separate the DNA. So to cut a long story short, when we do that, we see a nice product, we can use a sizing ladder to size that product, but really the key figure of merit here is the time. We're doing that complete analysis in around 15 minutes, and the reason that we can do that is because we're able to apply high voltages to do a very fast separation, and we're able to heat and cool our sample very efficiently. So immediately, this kind of device showed you that you could start to think about doing a complex sample in, answer out process, not in 5 or 6 hours, but in a few minutes. And so that really changes what the experimentalist, or in this case, the diagnostic procedure, can do. So that was during my time as a postdoc at UC Berkeley. And then when I came back to the UK, I was lucky enough to start working with one of the founders of the field of microfluidics, which was Andreas Mantz at Imperial College. And while we were there, we started to think about how we could use scale in an even simpler way to improve, in this case, the polymerase chain reaction. So you can see here, if we look actually, let's start off by looking on the right-hand side here. You can see here a schematic of our approach. And so up till now, if I just take maybe this bottle here, when I do a PCR reaction using a conventional system, effectively what I'm doing is I'm taking some reactants, putting everything in a container, and then I'm heating and cooling that. Now the problem with that is that it's going to take time to get heat in and heat out. The thermal mass is typically large. I may have variations in temperature. And this means that PCR historically was a very slow and often inefficient process. So we thought, why don't we do something different? Why don't we use the fact that we can make an extended channel? Let's do a space-to-time conversion. So now instead of having a static reactor, what I'm going to do is I'm going to push my fluid along an extended channel, And I'm going to make different regions of that channel set at different temperatures. And you can see how we did that. Incredibly simple. A glass chip with a single channel that goes backwards and forwards. We rested it on copper blocks, and we fixed the temperatures to our denaturation, our extension, and our annealing temperatures. Now, the advantage of this, again, is down to thermal mass. If you work out the volume of fluid, say, if you follow my laser pointer now, in a region corresponding to this 60-degree temperature, the volume is probably only a few nanoliters. And what that means is that it can almost instantaneously get to the set temperature. So that means our PCR reaction should be fast. And when we did the analysis in the old days using slab gels, what we found was that we could amplify DNA in times down to around 90 seconds. This was super fast. This was an order of magnitude faster than you could do using any other kind of technology, and actually starts to get very close to the limits which are going to be defined by the processivity of the polymerase enzyme. And all we've done is simply made the system small and moved our sample around. So technologically, the innovation is very small, but the result is very, very important. Now, although this was very nice, as with most science, there are always problems, and this last bullet point here is really important, is that although this worked and on paper worked very well, in reality we had lots of challenges, and those challenges come from the fact that we've got this large surface area to volume ratio, and that means that molecules see the surface. And this proved very problematic in many different applications, both chemical and biological. And so what I'm going to do for most of the talk today is talk to you about a format that we've used for lots of different applications, but particularly for biological applications. So as you'll find out very quickly, droplets are very close to my heart, and we can use droplets as really nice vehicles in which to do high-throughput chemistry and biology. So if we use bulk technologies to make droplets, they look like this. They're all different sizes, and if we were to watch a video or a movie of an emulsion like this, the droplets would start to merge together over time. So they're not really good formats to do quantitative biology. And so we were lucky enough when we started to have these problems to see some really seminal papers by Rustam Ismaghilov's group at the University of Chicago, where he started to show that you could use very, very simple microfluidics to make incredibly powerful droplet tools, as I'm going to call them. So I'm going to go through this slide very quickly, but then I'm going to show you some videos which I hope will really tell you very quickly why these tools have become very, very popular and very powerful in modern biology. So the idea here is, if we look at two little cartoons here, is that all I'm going to do is I'm going to bring an oil flow and a water flow, two immiscible fluids together, either at a T-junction or a little orifice like this, and spontaneously what they're going to form are plugs or droplets. And these plugs or droplets have lovely features. So the first one here is, as you'll see, they move at a constant speed. So what that means is that we can time processes very well. The chemical payload, what we put in them, can also be changed very easily. If you look at the cartoon up here, I'm making droplets here at a T-junction as it snakes backwards and forwards. If I change the flow rate of these individual streams going in, I can change how much of a particular molecule I'm putting in. So suddenly, I can make a whole train of droplets which are completely chemically or biologically different. And importantly, I can do that really quickly. So normally we would operate our systems making maybe 1,000 droplets a second or up to 10 or 20,000 droplets per second. So these suggest really nice applications in what I will call high-throughput biology. And so the kind of ideas that I'd like to draw out over the next few minutes are going to be using droplets to control processes, to confine things in, particularly cells, and to do things very, very quickly, and to extract lots and lots of information. So videos and pictures are always nice. These are just some videos of us making droplets. The thing to remember here is that these videos are measured with a high-speed camera. So in this case, we're making about 1,000 droplets, or at different frequencies, maybe 50 droplets to 1,000 droplets per second. And the volume here is between about 40 and 800 picoliters. And you can see here that simply by changing the flow rates in the system, we can change the droplet size. We can make super small droplets. So you can see here an example. Actually, this is for a very specific chemical application, but you can see here droplets which have got volumes of around 75 femtoliters, and we're making about 22,000 per second. And here, playing around with the rheology of the fluids that we're using, you can see different arrangements and shapes of droplets. But the important thing here is that we're able to make, particularly if you looked at these two examples, actually all of the examples, identical droplets. These are as close to being identical as possible. So within 1 second, we can suddenly make, in terms of volume, 10,000 or 20,000 identical droplets. And that then allows us to do different things with them. So as I said to you, droplets move, whoops, that didn't show it there, but droplets move at a constant velocity. So what I'd like to do is show you some examples of different unit operations. So when we started to make these systems, what we wanted to do was really mimic the kinds of processes that you would use in the lab when you're doing a conventional macroscale experiment. So I'm going to show you some examples of initiating reactions through merging, maybe encapsulating things, incubating things. And at all times, I think the most important thing to see or to notice is that we can do these processes in a very, very robust and reproducible manner. So the first example here takes a little bit of explaining, but what I've done here is we're making droplets, if you look here, a little T-junction. So I've got aqueous fluids coming in, I've got an oil flow coming down, and what we've done here is we've split the detection channel into two. So they're the same droplets, I'm just looking in the top at a green dye that I'm putting in, and in the bottom a red dye. And you can see here that this flow of red is constant, I don't change it, and so I'm dosing a constant amount of red dye into my droplet. In the top, you can see that this flow here changes width. And as it does, I dose different amounts of my green dye in. This is just showing you, just in a visual way, that I can change what we're adding to our droplets very, very easily. So we can change what's inside the droplets, but when we're doing an assay, and particularly in the work that we're currently doing, what we'd like to do is to be able to look inside a droplet, and if we see something we like, we'd like to move it out the way. And you can see here an example. Maybe if we look, let's just look at the top one. It's a little bit slower. We have a stream of droplets coming in. We have a detector at a position here. If it sees fluorescence that it's interested in, and hopefully something will happen soon, you'll see a flash here. We apply an electric field, and we take the droplet down. And so this allows us to pick out things of interest and do more complex processes. And so finally, two other simple examples, because again, I think they're instructive, is a device down here which allows us to initiate reactions. We're merging two droplets together, initiating a reaction and moving along. And remember, the time scale is short. The time between a droplet entering here and the droplet leaving is around 30 milliseconds. So this is occurring very, very quickly, but in an incredibly reproducible manner. And then a device here which does effectively the same as this device, but this is a device that allows us to take in an empty droplet and to form a droplet here where we can control the concentration. So this allows us to create concentration gradients which are particularly useful when it comes to doing things like high throughput screening. So these are all tools, but how can we use them in biology? And so I'm going to give you a few examples, certainly focused initially on DNA. So as I said to you, when we described continuous flow PCR, conceptually it was lovely. Very simple idea that gave you this huge improvement in performance. But unfortunately, what it does is it also gives you problems. And so a very simple way to get around this is to still use the power of flow, so to do this space-time conversion, but to now encapsulate everything in a droplet. So this is the device that we use to do that. This is very, very similar to the one I showed you, except now we put a hot pin at the center. So the center is hot, the periphery is cold. We move our sample in and out. And to see that more clearly, we can see a video. So now we're still moving our droplets, but now we're keeping them encapsulated within an inert carrier fluid. They don't touch the walls of the device, and what that means is that the efficiency of the process should be improved. And so there are a couple of nice features. So we did this a number of years ago. In fact, I think this is January 2009. This was a month after Darren Link's group at Raindance showed a very similar system. But what you can see here is that I know that my droplet volume is 130 picoliters. So when I do an analysis of the products, all I've done here is broken the emulsion once we've finished, analyzed what we've got. We see that we can detect template molecules here down to around 4 femtomolar. So if I know my droplet volume, I can then back calculate, first of all, how many copies of DNA I would have had in each droplet. And it turns out that in this case, if you were to maybe pick 3 droplets in a row, just one of them would have contained a single copy of DNA. So we're doing single copy PCR here. The carrier fluid is preventing any interactions, and so the efficiency of the reaction is much, much higher than we could get in a system where the surface is existing. And so this kind of technology has been now developed by many others, but really allows you to do sensitive PCR, it allows you to do digital PCR, and really this kind of technology is a key component in most next generation sequencing workflows. So very, very powerful. So the other thing that we did very early on, and perhaps is the most interesting in terms of where the biology has gone on to nowadays, has been to use droplets as simple ways to trap single cells. And the way we do that, again, is very simple. So I'm just going to use a T-junction again. I'm going to bring in a solvent, in this case a growth medium, and a cell suspension. So if I vary the flow rate of that cell suspension stream, I'm going to dose in different numbers of cells. And so the idea is in this very initial experiment, we form our droplets at this point, they move through the system, and then we have a little optical detector that looks at what's inside the cell. And actually, if I'm going to describe the data, I should show you how that works. This isn't in a T-junction, this is in a flow focusing junction, but what it shows is very instructive. I've got a stream of cells here, and then occasionally when I form this droplet, I encapsulate the cells. But what's perhaps most important, and if I can just hold the video here, is to look at what we've created. This is here a droplet. This maybe has a volume here of maybe 20 picoliters. What's really nice is that our cell, although it's smaller, it isn't that much smaller. And so what these droplets do, they act as perfect environments if we want to break open the cell and do something with it, because as soon as I break that cell open, the contents will come out, but they're going to be encapsulated within this picoliter volume. They're not going to dilute by 6 or 7 orders of magnitude, which they would do in a normal microfluidic system or a normal macroscale system. So droplets provide a way of harnessing and keeping the contents of the cell contained, keeping the concentrations high, and allowing us to do lots of different experiments. So just to show you, when you do this kind of experiment, you can see here these low square features. These are the droplets. The growth medium is fluorescent, so we can pick those out very easily. And the little spikes that you can see here are, in this case, E. coli cells expressing YFP. And by changing the flow rate, we can then change the number of cells that we're putting in the system. So you can start to apply this technology. It's not perfect, and I don't have time today to explain the implications of Poissonian statistics. I'm just going to show you, though, a few examples of how we can use this, or one particular example before I move on. So this is an example of an integrated workflow. So like I said, my job today is to kind of show you that by using these different tools, you can start to think about doing experiments in completely different ways. And this is a nice example where we're trying to screen photosensitizers for photodynamic therapy of cancer. We want photosensitizers that are going to be good at killing cells, and we'd like to screen for that. So what we do here in this cartoon schematic is we take a bunch of cells, we bring in a stream of photosensitizer, We can change the identity of that, so we can change whatever we're putting in, and we can change the concentration. We form droplets where we've got photosensitizer and cells in, and then what we do is we dump them into a reservoir. And in these experiments, we make maybe about 500,000 droplets. We can then expose this reservoir to light, so hopefully the photosensitizer is going to do its job and it's going to start to kill the cells. To assay that, what we then do after a particular period of time is we take the droplets off. In this case, we space them up a bit. We bring in other droplets which contain our viability assay agent. So in this case, we're using carboxyfluorocene here to report live cells, propidium iodide to report dead cells. We use our little merging unit to merge those together. We stick them in another reservoir to incubate so the chemistry can happen. And then at the end, we have our little laser-induced fluorescence detector. We probe the droplets as they go through. We can then pull out the raw signals here to report live cells, here to report dead cells, and then we can build up this lovely survival curve. So the reason I'm showing you this in this case is not particularly for the biology, but really to show you that you can start to integrate processes in a very robust manner. And so the experiments that you start to do become very precise, but also you can do lots and lots of them very quickly. In this case, maybe over an hour, you've done 500,000 different experiments. So what I'd like to do now, for the remainder of the talk, is we'll come back to droplets, but is to talk about a more general issue in microfluidics. So I'm hopefully doing some kind of job in telling you why these systems give you advantage. But we have a very obvious problem that as soon as I start to make something small, detection is going to become harder and harder. And so this is a slide which I won't go into detail, but it shows a very obvious feature, and that is if I have a cube, and I have a side of the cube going from 1 millimeter here down to 1 micron, the volume of my cube goes from 1 microliter down to 1 femtoliter. If I imagine I've got sample inside that cube at a particular concentration, here's say half a nanomolar, what I find is if I do the maths, I find here that as soon as we're down to a volume or a cross-section of 10 microns, which is roughly the kind of dimensions of the devices I've been showing you, I've got very few molecules in the system. Maybe 1 or 10 or 100 molecules in that cube. And that means, very simply, we have to be very good at detection. And so I think the biggest challenge in the field of microfluidics, particularly for biological experimentation, is here. Microfluidic systems are really good at doing things very well, but what we need to do is ensure that we're equally as good at extracting that information, and that's by developing really useful optical tools. And so what I'd like to talk about is one tool in particular. We've developed a whole variety of tools in the group, but one tool in particular developed by Stavros Stavrakis in my group, which really allows us to start to do what I would call massively parallel experimentation. And so here, I've got a simple statement. Extracting information is hard. And I'm going to give you one example of that, and that comes from droplet microfluidics. So as I said, we learned a lot of our early droplets from publications from Rustam Ismaghilov's group. And this was one of my favorite publications at the time. He showed that you could use droplets to extract enzyme kinetics. What you're looking at here is an image of a droplet-based experiment. And so the first thing, or the only thing that I want you to see from this when you look at it, is I can't see any droplets. And the reason you can't see any droplets is the droplets are moving too quickly. So because he can make lots and lots of identical droplets, he can still extract the kinetic information just by looking at the intensity of the product fluorescence here and turn it into a curve. But what he can't do in this example here is have lots of different droplets with lots of different conditions doing lots of different experiments because we've lost them. And so it's just like this image here. If you take an image of this London bus, you look at the foreground and the background, everything is nice and sharp, but our bus is moving too quickly with respect to the frame rate of the camera, and so we lose that information. And so that's one of our biggest problems in droplet microfluidics. And what Stavros did was realize that the answer to the problem had been solved decades before. And so, again, even though this is a biology-focused lecture, I'm sure you've seen images like this before. These are incredibly famous, significant images made by Harold Edgerton and his team at MIT back in the 1950s and the 1960s, where he developed stroboscopic photography. And the key idea here is that we're able to see these beautiful events, this bullet going through a playing card or a bullet going through an apple here, and we're able to see everything with high spatial resolution, not because the shutter speed of the camera is short, but because he's illuminated the scene for a very short period of time. And we're going to use exactly the same concept to look at droplets. So now, if our shutter speed is 10 milliseconds, we can create an effective shutter speed by synchronizing an excitation pulse, which is much, much shorter. And so we've used this to do a number of different things. And I'm just going to show you two examples, which again, hopefully will give you ideas about how this kind of technology can be used. So what we're going to do in this experiment is to do an enzymatic assay. So we're going to look at a particular reaction. This one involves formation of a fluorescent product using beta-gal as the enzyme. And what we're going to do here, if you can see in the schematic, we're going to bring a buffer stream here, and we're going to mix it with a bar-coded substrate stream. Now, the word barcode is important because what I'm going to do is I'm going to modulate the flow rate of this stream coming in, which changes or varies the concentration. And when we do the analysis, I want to know what concentration I had. So I put in a little barcoding die, which is going to tell me that. I then mix in my enzyme. I push it through a droplet generator. That moves along a windy channel. That allows us to mix things up very quickly. And then along a serpentine channel. And by using stroboscopic illumination, we're going to try and freeze that information and pull out the kinetics. And we do that using this detection scheme here. I don't think that's important to dwell on today. Let's just look at the data, and hopefully you'll see very, very quickly why this is so powerful. So if you were just to take an image, this is an image of droplets using our continuous illumination. We see nothing. We just got a high saturated signal. The droplets are moving too quickly for us to look at. As soon as we used our stroboscopic illumination, we can freeze the motion of the droplets. So we've done the first thing. We're able to see where our individual droplets are. We can then use the signal intensities to extract our kinetic curves. So you can see here an example of some raw data where this black curve here is my barcoding dye concentration. So that's telling me how my substrate concentration changes. And then this blue envelope of curves is my product concentration. So what I can do from this raw data here is I then can start to create some kinetic curves. In this case, we're just doing some simple Michaelis-Menten kinetics. Like I said, with this beta-gal assay generating resirufin, we can pull out the Km, but the most important thing here is that bottom bullet point. We've done 1,500 completely different, so these aren't repeats, completely different experiments in about 10 seconds. So this, using any normal technology, would take us weeks or months to do. By using this very simple idea, we can start to do really, really high throughput experimentation and generate lots of information. And you can do lots of other things like inhibition kinetics. And so really just to finish on two examples of this, at the end of my talk, I'd like to very nicely talk about two members of the audience here today. So Zbigniew Prokop and Iji Damborski, who are our close collaborators, and have allowed us to use with them this technology to start to do very important biological measurements. Now, I'm not going to focus so much on the importance of the biology, but just to show you how we do the experiments, I think is very instructive. So what we've got here is a cartoon which shows a very similar system. I'm just going to highlight a couple of aspects. What we're doing here is we're forming droplets. You can see them there. We're going to vary our substrate concentrations and our enzyme concentrations, but we want to look at very, very fast kinetics. And so what we do is we use these turns to create something called chaotic advection. This allows us to mix things very quickly, and we accelerate the droplets as fast as possible. So this allows us to bring in reagents and mix them in really short periods of time. I'll show you in a second how short. And then what we do is we then try and slow the droplets down, and we use Stavros's stroboscopic illumination to then start taking pictures of these droplets and then extracting our kinetic information. So how quickly can we do this? Well, this is an example here of just some calibration work where we're using a calcium-sensitive fluorophore here to assess our mixing time. So we add in this fluorophore, it mixes with a stream of calcium, we separate it with water so that we only start mixing when our droplet forms, and you can see here that our mixing times are incredibly fast. We're mixing reagents here in around 1 millisecond. So this has done a perfect job at mixing everything together, and then, like I said, we can use the stroboscopic illumination to do the kinetics over extended periods of time. And it turns out that this becomes a really powerful environment. So these are just 4 examples here of model enzymatic reactions using, in this case, horseradish peroxidase. Here are microperoxidase, beta-gal as well. All I want you to see really here is the timescale. We're looking at kinetics on timescales that are incredibly difficult to access using other technologies. And we're doing lots and lots of experiments per unit time. So here, over about 20 milliseconds, here 40 milliseconds. And in this case here, we're able to extend our timescale so that we can look here at a fast formation process of an intermediate, but then also look at its subsequent oxidation. And again, we can control that through the use of the microfluidics. So I'm not going to embarrass Iggy or Zbigniew by giving very bad explanations of the biology, but what we're able to do here is we're able to use this kind of technology to start to look at the mechanisms of enzymatic action. And what the Bernot group has done beautifully over the last few years is started to look at this class of dehalogenase enzymes where, if you look here at the wild type, we can start to engineer structural modifications. By putting a tryptophan, we can change here maybe the tunnel width. We can introduce new access tunnels. And by then doing both kinetic and thermodynamic experiments, we can pull out enormous amounts of information on short timescales, and as it says here, start to understand the mechanistic action of these enzymes. So again, we're using this very simple, well, in a sense, simple experimental tool to infer biology, but also remembering that we use good computational tools as well. So for my last few slides that I have to talk about today in any detail, what I wanted to do is completely move away from droplets, but use the same ideas that I've just introduced to think about how we look at cells. And I'm sure those of you who are experimental biologists have used this technology or had data extracted by this technology before. So flow cytometry is really the gold standard method to enumerate heterogeneous cellular populations. It's incredibly simple in the way it works. You take a bunch of cells, you push them into a single file, and you do that normally by using a sheath flow. And when they're in a single file, you have a laser and a detection volume, and you can generate fluorescence and scatter. And that gives you valuable information. So as it says here, forward scattered light tells us about surface area or size. Side scattered light tells us about morphology, and fluorescence can tell us about surface markers or different processes. Now, flow cytometry is lovely. You can really look at quite large numbers of cells per second. Normally, I would say between about 5 and 10,000 as a maximum. But there are lots of problems. And the problems are, as I'm sure many of you know, flow cytometers are really expensive. High-end cytometers will go up to $500,000. And so they're clearly expensive bits of kit, and they use large volumes of reagents. So they have some problems with them. So we started to think about how we could maybe use microfluidic tools to help the situation. And one thought struck us, and this is a development that a number of other groups have looked at as well. So if you think about flow cytometry, this is raw data, in this case from a microfluidic flow cytometer. But what this is lovely at doing, and what you can clearly see here, is I've got scatter and fluorescence signals on a millisecond time scale. I can look at lots and lots of cells, and I can generate things like this, these scatter plots, which tell me broadly, once I do gating, about different populations or subpopulations within a larger sample. Now that's really nice, but if I look on the right-hand side here, you can see almost the opposite situation. So now, I'm not looking at 10,000 cells, I'm looking at what, 15 cells? But instead of numbers, I've just got information richness. I've got this spatial information, I'm looking at different colors, different intracellular components. And so normally the biologist takes one of either course. They either take flow cytometry to gain throughput, or they go for microscopy to generate information content. And so we decided that we'd like to combine the two. And so I'm just going to show you a couple of examples of how we've done that. And again, using good optics and good detection, how this can give you really nice advantage. So this is really the final thing that I'd like to talk about today. So this is our Generation 1 imaging flow cytometer. So first of all, you can see it's a tiny device. So it's a little microfluidic device made in a polymer as a single layer. It consists of 3 components. We introduce our cells here. We distribute them using a little flow manifold. They then go through these 6 parallel channels which have these bends and turns in them. And those bends and turns allow us to control both inertial and Dean forces within the system. And for us today, what that means is that we can start to align the cells to a particular axial position within the channel. I'll show you what that means in a second. And then finally, we have this bunch of switchbacks here, which means that as our cells move out of these channels and go backwards and forwards, we're going to use our stroboscopic illumination to image them. So just to look at this in a cartoon, which gives you a little bit better idea, the sample comes in here on the left-hand side. You can see all the cells are randomly across the channel. By moving through these turns, we align them into a straight line, and then we're going to image them here. And actually, in the first experiments that Stavros did, this is the kind of image that we're pulling out. So we're measuring around 120 frames a second, and this is within this parallel section. And so if you start to analyze the data, you can see immediately where the big gain is. So maybe if we just choose one, let's just choose this here. What we're doing here is we're looking at a mixed cell population, HL60, K562 cells. By using scatter or by using multicolor fluorescence, we can pull out the information, and you can see here the rate is around 100,000 cells per second. So we're not just beating normal flow cytometry in terms of throughput, we also get the spatial resolution. So you can see here, this wasn't perfect when we did the first experiments, but this is when we image cells and they're static. We're not moving them at all. And then this is when we're moving the cells at a speed of around 35 centimeters a second. So you can see that the stroboscopic illumination really freezes the action. And then we can apply it to different problems. So here, we're looking here at multicolor fluorescence. This allows us to size our cells in terms of the overall cell size in the nuclear area. I think on the next slide here, we show that you can do more interesting things like cell cycle analysis, where we can start to discriminate between here the G1 and the G2 phase within the cell cycle. And when we try and do that using a high-end flow cytometer, we just can't do it. So rather than focus on this, I'd like to just show you where we are with this now. So this is what I would call our Mark II version of our imaging flow cytometer. So we use the same ideas, but now, in fact, we don't use inertial forces alone, we use viscoelastic forces. And what this allows us to do, simply by dosing in a polymer, in our case polyethylene oxide, at low concentration, it allows us to control the viscous and elastic forces within the system, and it allows us to align our cells. And so to cut a long story short, what we can do using a simple single channel that goes backwards and forwards is we can start to image at really unprecedented throughputs. You can see here an example of looking at some Jurkat cells here. We're using a frame rate of around 9,000 frames per second. And you can see the throughput is now around 400,000 cells per second. We're using the microfluidics to control the cellular motion and the occupancy within the field of view. And the throughputs become extraordinarily high. But we use fluorescence much more, so we can do an equivalent measurement at the same time. So you can see here a bright field image of 2 cells, but at the same time we can extract multicolor fluorescence. And what the team were really good at doing was showing then that you could start to look at intracellular components. So you can see here examples, and I forget whether this is yeast or human cells, but you're effectively looking at little RNA aggregates here. These are P-bodies or stress granules. And you can see here that we're measuring the cell, or we're looking at the cell, with a resolution which is actually close to the diffraction limit. This technology is super powerful, and what are we doing with it now is, well, we're trying to use it in diagnostics. So because we can create our system not for $500,000, but for about $5,000, you can start to think about a system which is maybe the size of a soccer ball, something like this, being used in a diagnostic workflow where a clinical sample like blood comes in. We can then digitize the images in real time, use machine learning, and we started to do this in a paper that will shortly be out in Cell Reports, where we're able to, in this case here, look at diluted blood, able to probe, in this case, a disease called Sézary syndrome, which causes changes in the nuclei of lymphocytes. It gives them this cerebriform kind of shape. By using our flow cytometer to generate images, and then using machine learning to characterize the digital signatures, we can start to call very, very nicely in real time or diagnose these kinds of samples. And this was something previously that would have to be done by a physician, literally looking through a microscope and making some kind of decision. So we're very excited about this. Now, I slightly lied, and I don't know how much time I've taken, but I do have 2 or 3 more slides, and these are just going to be flash slides, because in a talk like this, you can talk about some things, but on everything. But I thought it would be nice to show you some of the other things that we do. So like I said, I won't go through this in any detail. We can use microfluidics to control things. We do a lot of work where we house or we farm nematodes, C. elegans here. And by using microfluidics to confine the animals over the course of their lifetime, we can do high-resolution imaging and really extract a lot of information. So here you see examples of images where we're looking at seam cell growth in C. elegans, going from the early larval stage all the way up to the adult stage. And here, using no droplets, using no flows, but pneumatic systems. We're also using pneumatic systems to start to do other things. And again, the last 2 slides, I think, are unpublished data, but I'd like to share it with you today. So here, with our good collaborator, Rudy Aberzold at ETH, we're developing systems to start to do single-cell proteomics. The video here is a little bit faint, so I'm not sure you're going to see this, but using pneumatic microfluidics, where we're using effectively pressure to valve and to push fluid, what we can do is trap cells in what we call V-valves. So if you look at the video here, we take a little cell, we move that cell to this trap here. It's a leaky valve. We hold the cell there, and then we can deliver that cell into a little lysis chamber, and eventually, you can see the cell there. We can then lyse the cell. We can deliver that to a digestion chamber, and then we can do mass spec on the sample. And with all the recent advances in mass spec, what becomes really super cool is that you can start to pull out enormous amounts of information from the mass spectra. So here, from a single cell, we're able to extract signatures well in excess of 1,000 proteins. And so this is a really exciting area for us, but you can see using completely different microfluidic structures. And then as a final biological application here, again, something which I know is of interest to many people here in Brno, is the isolation of things like exosomes and extracellular vesicles. And what we're doing here is using those viscoelastic tools to do passive separations. In this case, all we have to do is deliver a sample through this reservoir at 2. The sample goes through the device in this direction, and then at each of these outlets, whether it's A, B, or C, we can detect different fractions. So here you can see here we're looking at particles which range in size from 1 micron down to 100 nanometers. We can perform very nice separation, as you can see in these images here, and we apply that idea then to blood, where we take a sample in which is predominantly blood cells, so white, red platelets, with a very small number of labeled extracellular vesicles, and by simply pushing the fluid through that device, we end up here with a system which has 96% of the extracellular vesicles and now really less than 0.2% of the blood cells. So again, this technology I think is lovely because of its simplicity. We're making use of scale, we're making use of rheology to affect something which is really difficult to do normally using other tools. So at that point, I have come to the conclusion of my talk. So I think it's always important to try and leave with some kind of message. And I think that message is pretty clear, that very, very simple microfluidic tools, all the examples I've shown you today are not complex in their fabrication, they're very simple, can almost always be used to improve efficiency, control, functionality, and throughput. But there's a caveat, and I think that caveat has been borne out in the last part of the talk, is that we need good optical detection methods and nowadays good machine learning. And what that really offers us is a route to doing incredibly high throughput experimental biology. And I do think that if Mendel was still around now and he had some of our microfluidic tools, he would have put them to very good use. So with that, this is always the most important slide. I'm the spokesman. I have the lovely job of telling you what we've done. The work is done by others. In this case, I can't name them all, so this is just the current group. A particular highlight is Stavros Stavrakis, who's sitting in the front row today, who developed a lot of our optical detection techniques. And then finally, the people who gave us the money. So with that, I thank you for your attention and would be willing to take questions if we have time.
So, thank you, Andrew, very much for this fascinating tour in the world of microscale experimentation. And now I'm opening the discussions. Questions, please.
Great lecture, thank you.
Thank you.
I was wondering whether you are thinking of using your technology in structural biology, so actually using these droplets, identifying individual proteins in numerous conditions and trying to combine them with negative staining or cryo-EM actually to identify groups of, you know.
Absolutely. That's not anything that we've done, but you've effectively defined how you might do that using a droplet workflow. And I think the most important point there is that using the statistical ability to load single entities within to the droplets, you can start to really pull out huge amounts of information. So we've been starting to get interested in things like crystallization, both in terms of protein crystallization, but also in inorganic crystallization as well. And I think that kind of aspect is really important. So you could easily think about those kinds of applications, which people are doing.
And the other question would be, so is the detection limit, let's say, if you would want to look on the individual protein reactions, single molecule-like, so in the fluorescence scale, is this something that you can monitor, depending on using dyes?
Absolutely. So, I mean, I'm old enough to remember the first time I read a paper about doing single molecule detection in condensed flow and thinking this was crazy. All the developments in detector and light source technologies really allow us to do that relatively routinely now. So pulling out the information is not the problem. The problem typically for single molecule detection has always been you can detect single molecules in the condensed phase, but you're wasting most of them that you never look at. Now, one of the really nice things is that as soon as you start to use droplets, you confine them so your molecular detection efficiencies shoot up. And so you start to look at every molecule potentially in a volume. And this is going to be really important when you think about things like circulating, tumor cells and things like this, where it's not just a case of detecting something, it's ensuring you've looked at everything else and not missed anything as well. So, yeah, we love doing optical detection. So, many things like in the example of the mass spec are great because they give you so much more. But one of the beauties of using optical techniques is you're able to pull that information out so quickly.
More questions? So maybe I will ask, Andrew, where you see the major challenges of the field and what to do in future to bring these fantastic technologies to broader biology community?
I mean, how long do I have? No, I mean, there are many, many problems, and as I'm sure many of you know, or many of you will learn, when you give a talk, you present the successes and you distill maybe huge amounts of work. There are many problems in the field. I think materials have been a big issue. So the material that I've used for most of the devices that you've seen there is a material called PDMS. It's silicone polymer. It's what's typically been used in breast implants. It's beautiful to work with, very, very cheap, very easy to make structures. And it's very good for certain biological applications. For many other applications, it's terrible. It absorbs hydrophobic molecules. It causes lots of different problems. And in fact, Zibignak's asking me the question, in the work, our collaborative, the work that we spoke about in this example, just shooting backwards here, we used PDMS as the format for our initial system, but because of all the issues, we then had to develop really smart coating technologies which allowed us to do all these experiments that you see here. So materials are still a problem. You have to think of the right material for the particular process that you're looking at. And I would also say it's very obvious. Microfluidic systems are not a panacea. They're not going to allow you to do every experiment in the best way. So you have to think, where does the system give you advantage? And in many, many examples, going to a complex microfluidic system wouldn't be challenging. I think, however, what's been really nice to see over the last few years is in any field is you start off with lots of examples. We can do this, we can do that. And then the field stagnates a bit. I think what people are really doing now is using microfluidics as a basic tool. So I think we were talking today, I think the field has become a success when the next paper that Zibignak writes, the microfluidics is hidden in the supplementary information. It's not the star of the show. It's simply a means to an end. And I think really that's where, a lot of these tools, particularly things like droplet PCR and single-cell encapsulation techniques like DropSeq and InDrop have really changed the way that you do RNA expression profiling. These technologies really use microfluidics as a basic tool. So I think we're getting there. But there are still so many things to do. So lower concentrations, higher throughputs, more information.
Right. Thank you. More questions?
Thank you for a very nice talk. I have a question regarding this technology which could potentially replace fax. Do you think that might happen eventually, that because of price and advantages, we will see replacement of fax?
I mean, again, you always get excited when you do something in a group, when you see what your team is doing. For me, the arguments to use microfluidic imaging formats to do flow cytometry are completely compelling. We really beat standard flow cytometry formats hands down very easily. There's always inertia, as you know, with technology. It takes a long time for technology to really impinge. What we're doing now, and I think is really the biggest testament to how robust the technology is that we're using the Mark II version of that flow cytometer I showed you in clinical settings now. So we've been building instruments which are now being used at the University Hospital in Zurich on real patients, on clinical samples. And I think and they work beautifully. So what I would like to see is moving away from this idea that flow cytometers that are good have to be in centralized labs that you send a sample to, And you now start thinking, well, I have my own flow cytometer, and it will do everything that the big ones do. So I absolutely believe, not just our technology, there are a number of other groups who've developed other technologies. Something I didn't get to talk about today, which again we're very excited about, is using deformability cytometry. Can we control shear and stresses within a microfluidic system to distort cells and use the deformation as a phenotype for different diseases? And that, again, works beautifully well. So I think there are so many different possibilities in that respect.
More questions?
Yeah.
So what is also intriguing, this lab-on-chip technology, so when you're really trying to combine multiple methodological approaches, so how do you see the future of this? You know, ideally, you know, expressing protein, purify it, then maybe sort it, look from the different angles. What is the modularity of this system, you think?
So,
in the near future,
two great questions. Okay, so to answer that one, we use an analogy, and I think the reason I showed you that example of a photosensitizer assay was that we put a load of things together and it worked really nicely. And if I'm an electrical engineer and I design a new circuit, I don't build that circuit to test it. I test it on a computer. And if it works on the computer, I know it works in reality. Microfluidic systems are completely different because everything is coupled. And things work beautifully well on their own. As soon as you put things together, it creates challenges. So I think it really depends on the application. There are many times, again, talking in discussions today, where we've been trying to make, without going into detail, sort of cellular mimics where we're creating membranes around droplets. And to do this has proved incredibly challenging. We've got a recipe that works now, but it really took a huge amount of time to get things going. And as soon as you start to daisy chain more and more different processes, it becomes problematic. But there are many really good examples. So I would say you can build the lab on a chip and you can increase the complexity to where it gives you the most advantage. But that may not be the whole process. It may be only a component part of the process. But I think, and the reason, you know, I've spoken about droplets today, and the reason we've used droplets is they are robust. So when I was young and I did experiments in the lab using continuous flows, it was terrible. Small pipes doing chemistry and biology, and they block after a few minutes. And so what you were doing is showing things might work, but there were too many practical problems. With droplets, you really start to create these robust systems that work not for minutes or hours, they can work for days or months, weeks and months over different processes, because you are really isolating the interesting stuff from the surface. So I haven't really answered your question, but integration is really there. So there are many groups, much better than our groups, who've really shown highly integrated processes, and they do work. But I would always work out the level of integration with respect to what the advantage ultimately is going to be.
More questions? Maybe I have one.
Okay.
You had this parallel in the electronic industry where we have this exponential Moore's law, but actually it would be not exponential but sigmoidal at the end. And we now approaching. So, because these molecular restrictions of the isolation of the layers.
Exactly.
And there is a solution in computing, quantum computing.
Mm-hmm.
Do you see any future solution for microfluidics when we will hit such a similar limitation if we keep this power?
I mean, that's really interesting because I think in a sense, if you use Moore's law for an electronic circuit, the smaller, the better. Let's make it smaller and smaller and smaller and smaller until we can't make it any smaller. In microfluidics, that doesn't quite work because of practical constraints like we were just talking about, maybe precipitation or blockage. So I think a really good sort of working method is that you make a system smaller and smaller when it gives you advantage. Or let's do it the other way. If a system works beautifully, you can make it bigger and bigger until it stops giving you that advantage. And that's probably where the right dimension is. So I don't think there are going to be some examples, you know, people who've developed things like nanopore-based sequencing clearly need these very small dimensions. But with the kind of systems I've spoken about, you actually want to stop. You don't want to keep on getting smaller. So I think more of the challenge will actually be, how can we create systems which integrate all the control architecture, all the detection, all the data analysis? That's more of a challenge in terms of increasing the power, rather than just making everything smaller and smaller.
Definitely. Thank you very much. So, is there any more question? If not, Andrew, thank you very, very much for accepting the invitation and
pleasure.
for this fantastic talk. Acknowledgement for your great science and coming here to visit us and share with us. There is a medal with the Mendel.
Oops. Put that there. I was at a prize given recently, and the awardee dropped the medal 3 times, posing for a picture like this. So, is that good? Thank you very much.
Thank you very much.
Thank you, Will. It's lovely. Beautiful. Thank you. Thank you.
And thank you very much to all of you coming here, join this lecture, and restarting the Mendel's lectures again. Thank you very much. Have a nice evening.